you. So I'd like to start out with this slide, which is a slide of the blood willow, the Salix flavophylla. It's an Alaskan willow, and it's, I guess also next year we'll have our meeting in Alaska, so we can all go enjoy blood willows. It's a little dwarf willow that I'll be talking about today, in part. So sex determination obviously is super important for during the development of an organism, especially dioecious organisms that have males and females. And that sex becomes associated often with sexual dimorphisms. And sexual dimorphisms are, common throughout animals and plants, as you can see here in this Silene latifolia. Also, sex chromosomes develop that sometimes become different in size and heteromorphic, and the evolution of those sex chromosomes is integral to understanding a lot about genomic evolution. Let's see. So, given the importance of a sex chromosome, you'd think it would stay in the same place, that it wouldn't be moving around, because it becomes linked with all these other traits that are important for mating and for fitness. But what we see in a lot of animals is, in some animal groups at least, is that the sex chromosomes move around. So this is an example from cichlid fish, where these are different genera of cichlid fish that occur in Lake Malawi and some other lakes in Africa. And they're actually, the sex chromosomes, or the sex determination region, are in 13 different chromosomes. And it switches between XY and ZW. Now this is completely different than what goes on in mammals. We know that there's really only one location for a sex chromosome in mammals. So why do we have these situations where the sex chromosomes move around, and then there are other situations where they don't? This is one of the big questions underlying our research. So I'm just showing here is a phylogeny of angiosperms with all the angiosperm orders. And what I want to talk about is that plants are actually a really great model system in order to study the evolution of sex chromosomes and the evolution of sex, or different sexes. Although only 5 to 6% of all plant species are dioecious, and few have cytologically visibly different sex chromosomes, there are many different evolutionarily independent events in which dioece has evolved from hermaphroditic ancestors. It's also hasn't really been determined whether plants exhibit the same dynamism in some time in these systems as animals do as far as the movement of the sex chromosomes. So to understand the movement of sex chromosomes, you also need to identify a group of plant species that has many species that are all dioecious. And that's also fairly rare. Often you find only one or a few species that are related to one another in the small clades that are all dioecious. So we've been working on the Salicaceae, which are the Malpighiales here. And the Salicaceae is a group where almost every species in the whole family is dioecious. And in particular, we're working on populus and willows because they're all dioecious in their sister genera. I'll just give a short introduction to these two genera. Populus is 30 tree species. These are all wind pollinated. Salix has about 400 to up to maybe 1,200 species. They're primarily insect pollinated, and these can be trees, shrubs, or like these dwarf blood willows here, like I showed at the beginning of the talk. They're all woody and fast growing. They're all dioecious. There we go. Thank you. They're all dioecious. And they also all have 19 syntenic chromosomes, and so we can use one species to map onto the other. And they all have relatively small genomes, and so that also makes it useful for population genomics and other genomic studies. So I'm not really going to talk about this. I just want to show it briefly. This is a phylogeny that we've created for Salix and Willow. It's based on it's a species tree based on 951 gene trees, and it was analyzed using RAXML and Astral 3. I can talk about this later if anyone's interested, but the main point I want to bring up here is that this group is Populus, this group is the Willows, it's Salix, and these are sister genera. The Populus, the sex chromosomes are primarily on chromosome 19 in Populus, and most of the systems are XY. Their Populus alba is a ZW system, and the sex chromosome is also on chromosome 19. In Willows, however, the sex chromosome, in the few species that have been studied, has always been found to be on chromosome 15, and all of those species are ZW. So we know at least between these two genera, there's some movement of the sex chromosome, or perhaps maybe diece evolved twice, and that's why we have two different sex chromosomes. And so we're interested in those questions. Primarily today, I'm going to be talking to you about willows and what we've been doing about mapping sex determination regions of sex chromosomes of willows. So this is just a pruned phylogeny, so you can see some of the species we're working on. I'll talk to you today about the most basal group, which includes Salix nigra, which is a tree species, Salix exigua, which is in subgenus Longifolia. These have these very thin, long leaves. And these grow around here as well, throughout the southeast, or I mean, sorry, desert southwest. And also talk to you about Salix phylogeny. And finally, I'll talk a little bit about Salix purpurea, because some colleagues and collaborators of ours, Steve DeFazio, who's a co-author on this talk, has mapped the sex determination region to be chromosome 15, and this is a ZW system, just like I was talking about before. And he's identified, their group has identified that the sex determination region is in a 7 megabase region on chromosome 15. And there's actually a inverted repeat there that makes a small interfering RNA that we think is controlling sex. I'm not going to be talking about that today. The way we identify these different sex determination regions is basically we're looking for these differentiated regions of a sex chromosome. And so if it's an XY system, the male has 2 different chromosomes. And if it's a ZW system, the female has 2 different chromosomes. So the male would have excess heterozygosity in this region, and the female would have excess homozygosity in that region if it's an XY system. If it's a ZW system, the females would have excess heterozygosity in that region. So what we do is we genotype males and females that we collect from natural populations, and then we look across the genome for regions of excessive divergence. The way we can do this is we can either use some association like GWAS techniques, or we could just calculate FST between the male population and the female population and find out which regions are the most diverged. And so this is what I'm going to be talking to you about today. This is the statistic that I'll be presenting is FST. So we developed a sequence capture array that covers about over half of the exons in the genome, or over half of the genes in the genome, at least one exon and over half of the genes, 16,500 loci, and we genotype about 5.2 megabases of data per individual. And then all of this, for at least this study, all of these have been mapped to the Salix purpurea reference genome. So here's a result that was done by Brian Sanderson. And this is what's shown here is the FST comparing 24 males and 24 females for Salix nigra, shown here. It's a tree-form species. And so we have the 19 chromosomes here. Each dot represents the FST of that single nucleotide polymorphism site. And the ones in red are in the top 0.1% of FST. What you can see is, obviously, you can see this huge column of high FST sites that are all co-located. There's a few others here. We think those are just mismapped. But that region is the sex determination region. If we, oops. For some reason, it didn't make it. Well, if we zoom in on that region is, let me just say a couple things about that. So this is on chromosome 7. It's a completely different chromosome than on chromosome 19, which is Salix purpurea. Also, if we were to be able to see the next part of my slide, which is to zoom in on this, we'd see that it's much smaller than the sex determination region on chromosome 19. So I don't know why that's not showing up, but it isn't. This slide is really hard to look at, so let me just walk you through it a little bit. Each of these is just a different SNP in the sex determination region. And if we look at the heterozygosity of that SNP, the males have excess heterozygosity and the females have excess homozygosity, completely consistent with an XY system. And so those patterns are consistent with XY. So we think that for Salix nigra, this is on chromosome 7, and it's an XY system. I'll just be walking through all of these. This is for Salix exigua. This is some work done by Nan Hu, a graduate student in my lab. He's mapped the sex determination region to be on chromosome 15, because that's where all the high FST markers are. It doesn't show up on here. It's also co-localized with the sex determination region in Salix purpurea that Steve DeFazio has been working on. But the difference is that the males have excess heterozygosity in these, for these SNPs around this region, and the females have excess homozygosity, which is consistent with an XY system and is quite different than what's going on in Salix purpurea. So here we have a 15 XY system. And then finally, in Salix phobophila, where we mapped, where we compared FST between 22 males and 25 females, again, we find that sex determination is on, the sex determination system is on chromosome 15, and it's a ZW system where the females have excess heterozygosity and the males have excess homozygosity. So I want to put this all together on the phylogeny. Let's see, so already I've talked about Salix purpurea, which is chromosome 15 ZW. Salix sichuensis is another species that's been mapped by some collaborators of ours, Po-Ming Yin in Nanjing Forestry Institute. That's also chromosome 15 ZW. I told you about Salix phobophila, that's chromosome 15 ZW. Salix viminalis was mapped by a European group, that's also 15 ZW. If you were able to see Minghao Guo's poster last night, you would have seen that he's mapped sex determination in Salix reticulata and Salix nivalis, and both of those are chromosome 15 ZW. And so this whole crown group, which includes the subgenus Vetrix and the subgenus Chromatia, is all chromosome 15 ZW. When we come out here to Salix exigua, that's chromosome 15 and an XY system. Some colleagues of ours in China, Zhenchuan Liu at Sichuan University, has also mapped the sex determination system in Salix arbustifolia, it's also called Chaucinia arbustifolia, to be chromosome 15 XY as well. And so we think that some ancestry about this point had a chromosome 15 XY system, and then there was a shift in dominance from XY to ZW. Then moving on to Salix nigra, this is chromosome 7 XY system. Also, our colleagues at Sichuan University have mapped the sex determination region in Salix caneminoides to be chromosome 7 and also on XY, an XY system. So one hypothesis is that the ancestral sex determination region is chromosome 7 XY. The other possibility is that there was a different chromosome that was a sex chromosome in the ancestor, and somewhere along this lineage it's shifted to be chromosome 7 XY. And we're mapping Salix triangular right now to see if that will kind of help to resolve this problem. So future studies we're interested in is we're interested in whether or not what's going on here. Is there one gene that controls sex determination, and that single gene is moving around in different places? Or do we actually have novel sex determination genes that are evolving in different places and causing the sex chromosome to move from one place to another? We're also interested, of course, in why the sex chromosomes move. And we have some hypotheses about that and are doing some ecological studies to try to address that, but we really aren't along to that stage yet. So with that, I'd just like to thank all the ton of people who have done all the work along with me, including Steve DeFazio, Brian Sanderson, who was instrumental in a lot of this work, Larry Smart at Cornell. I forgot to mention that Steve DeFazio is at West Virginia University, Ken Kiefover-Ring, and Jen Schmidt-Liu at Sichuan University, and also Tongming Yin. So that's all. Thank you.